environmental protection plant environmental protection sciences in the Mark Wright's lab, and he will be talking. His research is on exploring biological control agents, mostly parasitic wasps of invasive bark beetles, especially focusing on invasive short hole borer and also ambrosia beetles of ohia trees that spreads rapid ohia death. And today he will be talking about parasitoids of invasive bark beetles in Hawaii. David, whenever you are ready.
Hi, everyone. I'll share the screen. That work?
Yes.
Okay. So thanks to everyone for being here, and thanks to Roshan for putting this together. My name's David, as Roshan said. I'm going to tell you about some new parasitoids of bark beetles we found in Hawaii and what we know about what they do. So what is a bark beetle? The general form of a bark beetle is in the picture on the right here. Almost all species tunnel into wood and live and reproduce in it. Examples of bark beetle holes in trees are shown in the middle two pictures here. The entrance to their tunnels are typically small circular holes about the size of their bodies. So if you see that, especially if it has some fine powdery frass coming out of it, it's a sign that they might be there or have been present in the wood. Bark beetles can generally be split into two categories. On the top left is a xylem feeding species, where you can see the beetle having tunneled into the heartwood, where it produces a fungal garden that digests the wood, and then which it eats as a food source. And on the bottom left here is a phloem feeding species, where the beetles live just under the bark in the phloem layer, which is a more nutrient-rich place because that's where the sap flows. In Hawaii, we have a really nice native radiation of bark beetles. They're all likely from a single common ancestor and now are about 21 species. They are almost exclusively found in native forests associated with native trees and are an important part of our native ecosystems. We also have a bunch of inventive species that have shown up on the islands, now about 39 species. Many of these have somewhat of a broad host range, meaning they go after a variety of different trees. Many of them typically go after recently dead wood, but others go after trees or plant parts that are alive or somewhat stressed, and these can be problematic. Here are the known troublemakers found here. There are a few species that take an interest in ohia trees that are affected by rapid ohia death. You'll hear more about these later on from Kyle Roy. They burrow into the trees, and they have been shown to excavate, along with the wood, viable spores of the ceratocystis fungi that cause rapid ohia death, which can become airborne or possibly soil-borne. Excavating these spores into the environment may be a component of how this disease spreads in forests. Next is the coffee berry borer. This beetle burrows into coffee berries and eats their insides, which is the part that us people would like to turn into coffee, and so they're problematic for coffee growers. The tropical nut borer is a related species with a large host range. It is especially a problem for macadamia growers, where it burrows into the husks and eventually the seeds, which are the macadamia nuts. The black twig borer is another species with a very wide host range. Here it is in coffee. It often goes after living wood, and what it typically does is go into the small branches. You might be able to see an entry hole somewhere over here if you look closely. And that results in the terminal part of the branch dying. And here are shot hole borers, which have recently been shown to be a complex of cryptic species, two of which are present here. They also have a large host range and go after wood that is alive or in a low state of stress. One species is even known to spread a fungus that can kill the trees. If you see bark beetles in your trees, it's often hard to know if they are the primary or secondary problem. That is, whether they are weakening or killing an otherwise healthy tree, secondary on a tree that is already weakened and is on its way out, or further weakening a sick tree that could possibly recover if otherwise left alone. Bark beetles are able to pick up on stressed wood often before any problem is apparent to our set of human senses, so it's often hard to tell. Sometimes they are known to spread pathogens, either by vectoring them themselves, by creating holes in the tree that allow for the entry of those pathogens. It turns out that bark beetles, like most other insects, have a set of natural enemies that eat them. Worldwide, these include other insects such as a variety of beetles, fly larvae, and bugs. It's often hard to confirm these relationships because the interior of wood is often a very busy place, and they typically find and feed on the beetles inside the wood, so seeing that happen is difficult. Identifying what they have been eating by DNA in their guts is one of the few ways to show this with any confidence. Birds, lizards, pathogens, and nematodes have also been shown to limit their numbers to some degree. Some species of parasitoid wasps, the group of insects that we will mostly talk about here, are also known to go after bark beetles. In general, the predatory creatures are more generalist. They eat many different things. Parasitoid wasps tend to be more specific to using certain species as hosts. Because of how they develop, they have to be more closely tied to the life history and immune system of their hosts. This host specificity is often an important quality for biological control because the natural enemy will go after only what you want it to control, and its population will therefore track the population of its host. Bark beetles are difficult to get at living inside wood, and there are relatively few natural enemies, especially parasitoids, that are known to use them. This is a graph showing relative numbers of parasitoids among feeding guilds. Left to right is more concealed. So on the left is parasitoids associated with hosts that feed totally exposed, such as crawl around on leaves. And here under the blue arrow is boring insects. There are relatively few parasitoids that are known to attack them, but they do exist. And so here's a few that we found in Hawaii and talk about now. So these are two Phimasticus species. The one on the right is Phimasticus xylobori, and on the left is a species that we're describing. They both go after bark beetles in the genus Xyloborus. They lay their eggs in the adult beetles when they're in their tunnels, and then a while later, chew this hole in the top middle picture in the back of the beetle to emerge into the outside. The picture below right here is what that often looks like when the beetle is still in the wood. Three of the species the new one attacks, which are Xyloborus ferrugineus, Xyloborus affinis, and Xyloborus perforans, are three of the 5 beetles that are now known to possibly spread the fungus that causes rapid ohia death. We don't know their effect on these beetles in ohia trees, but an octopus tree, they seem to have a high percent parasitism in some places. We've calculated 72% on the 3 known host species together. We wonder if there's a possibility that they could be released augmentatively to control these species and possibly limit the spread of the disease. We've also seen them developing inside beetles in the Eualacia fornicatus complex.
Join the meeting.
Inhabit octopus trees. It seems that this is rare, maybe just a mistaken identity with their more common hosts in the same wood, but it does happen, and the wasp
You muted yourself.
Sorry, David, can you unmute yourself?
Okay.
Sorry, I muted actually. I cannot find you. Yeah. Okay.
Okay.
Yeah. Sorry. Yeah. Somebody made
Sorry
noise,
about
so
that.
that's why. Yeah. No, I did it. Sorry. Yeah.
Okay. So there might be a third Phimasticus species that's here before too long. So the only other species that's known in this genus is a parasitoid of the coffeeberry borer that has been imported to much of the coffee growing world from its native Africa as a biological control agent. So testing of this agent has been done in Hawaii, and it appears to be safe. So it is possible that it may be introduced here in the future. So this is the undescribed species that was on the left. The female wasp lands on the wood and searches the surface for beetle holes. She's found one here and checks it out. And then she backs down into it, digging through the frass and debris. Once she gets in there, she'll stay down there looking for a beetle. This is fast motion now. And a little while later, she appears back at the surface going around in circles. She's actually riding on the beetle and parasitizing it. The beetle doesn't like it and is turning around in the hole, attempting to shake off the wasp. So here's a little while later, and look, you can actually see the beetle. The beetle is really trying, but it doesn't seem like it can do much about the wasp on its back. The wasp usually lays 2 eggs in the beetle, 1 female and 1 male. Okay, so a few weeks later, watch the beetle near the top middle. The wasps have developed and chew a hole in the back of the beetle to come out. In real time, this takes about an hour, and that's the female. She comes out and stays right there nearby. A little while later, the male comes out. He goes for a walk, which is probably to find a mate who's not his sister, but then he's come back, and because that's the only option. They check each other out, and then they mate. And then they both fly off. Here's the next wasp. It's a pteromalid in the subfamily Cerocephalinae and possibly in the genus Cerocephala. We've seen it in Ficus microcarpa and also in mango trees going after cryphlis beetles, these guys, which live under the bark. To see what they are up to, we peel off bark down to the cambium layer, sandwich it between plexiglass and aluminum, let the beetles go in it, and then they live their lives and make tunnels, as you can see here in this little picture. We release a wasp and it searches through the tunnels. Look at it turn around like that. The tunnels are often filled with debris from the feeding beetles, and this is how it moves through them. This is sped up to double speed, and it grabs pieces of debris with its big mandibles and uses its legs to push them to the back of its body, opening up space in front. When it finds a larva, it stings and paralyzes it. You can see here the ovipositor inside the larva. This process usually takes about 15 minutes, so here we're speeding it up. Then it withdraws the ovipositor, now back to regular speed. It's going to turn around to host feed on the larva. We're going to go back into fast motion when it starts feeding. And 2 things to notice are you can see the larva decreases in size and the wasp's abdomen distends and excretes a liquid, probably like mosquitoes or aphids do to concentrate nutrients within them. When the wasp withdraws its ovipositor, it does so slowly and carefully, and often kind of extrudes this sheath around it. It leaves this thing here sticking out of the larva. And so when the wasp feeds, as you can see here, it puts its mouth parts on it and sucks through it, kind of as if it has made its own straw. So these bark beetles build a hard pupal chamber around themselves before they pupate. This pupa looks like a good place to lay an egg. So here it plants itself, and then it pushes and pushes. It's just sped up. And then look at that. There's the ovipositor. It's broken through. It probes around and it gets into the larva. Now back into fast motion. This takes about 15 or 20 minutes in real time. And then it withdraws and will put its mouth on the same place and start to feed. When it does, we're going into fast motion again, and notice how the abdomen distends. So it really seems to be sucking the juice out of the larva and looking at the distance between them, seemingly through one of those straws it has made. Here's one more video. Look at the ovipositor, how the wasp probes and flexes it to search for the beetle inside. It then lays an egg next to the paralyzed larva or pupa, and the egg hatches, and a little ectoparasitic wasp larva comes out and attaches to the beetle larva. Over just a few days, it sucks the juice out of the beetle larva, transferring it to its own body. And then it leaves this empty shell that was the larva, and then the wasp pupates in the tunnel and turns into an adult. Just a note on this, so these boxes we're looking at, you can take off the cover and manipulate things inside and then put it back together without much disturbance. And it turns out that if you take the coffeeberry borer larvae and replace them with the cryphlis larvae, it will parasitize them and complete its life cycle on them. So here are 2 prorop species. The 3 pictures on the left and the 3 pictures on the right are the different species. They both seem to have a similar biology. They're both parasitoids of the adult stage of hypothalamus eruditus beetles, which is unique in a couple of ways. They're the only bathylids known to parasitize the adult stage of their hosts, and they're the only known ectoparasitoids of adult bark beetles. Here's how they develop. It's always similar to this as far as we've seen. They lay their egg on the ventral side of the beetle in a soft membranous area between the pro and mesothorax. You can see it right there. And then the larva comes out and feeds on the beetle through that soft spot, grows and grows, and then eventually it'll spin a pupal cocoon where it turns into an adult. These are pictures of wood taken from the forest after peeling off the bark. In the middle, you can see larvae developing on the beetles. Each one has one on it. And on the right and on the left is more feeding larvae and also some pupal cocoons. Interestingly, all the beetles in these pictures were either paralyzed or parasitized. So it seems these wasps sting and feed on or parasitize a big proportion of the host patch. Those last pictures were of kind of wider gallery systems, but when they go after beetles that are confined in areas such as a tunnel system, here's what happens. So given where the position where the larva feeds on the beetle, as it grows, it forces the two halves of the beetle apart. And here you can see the wasp position pupating between the two halves. It's almost as if they figured out how to use the sclerotized body of their host to protect them in a tunnel. By peeling off the bark, I disturbed them and broke open their pupal cocoons. So the larvae, aren't exactly where they were when originally, but you can see where they were by the position of the pupal cocoons. These wasps sting the adults, larvae, and pupae. Here's one stinging an adult hypothalamus eruditus beetle. Next is it turning around. It's a lot like the pteromalid before. They're not at all closely related, but they both have a long articulating prothorax that seems to help them bend and do these agile maneuvers in the tunnels. Here's it biting a larva. It's stung and paralyzed. It could just be tasting the cuticle of its host. Seemed to not break through. And then here it is host feeding on a paralyzed adult through the same soft spot where its larvae develop. And interestingly, we've seen it biting adults too. Maybe it's also tasting them or attempting to move them. So this is the other species, and here it is stinging a pupa. And then later appearing to host feed on the same spot. One characteristic of the genus Prorops is that they have a nose. It's a structure like this that kind of sticks out of their face. It's kind of a question. What's its purpose? It turns out what we've seen it used for is host feeding and oviposition. In this video, the wasp is going to move the beetle and then host feed on it. The beetle has this hard cuticle that it wouldn't be able to bite through, and it does so through the soft spot between the pro and mesothorax, the same place through which the parasitoid larvae feed and where the egg is laid. What it does is using the low spot between the nose and mandibles, you can see it again in the top right here, is push on the side of the prothoracic sclerite of the beetle to expose that soft spot. And then the face is kind of a tongue and groove mechanism, and it chews on that soft spot with its mandibles. So this is it going to oviposit or lay an egg on its host. First, it does a lot of preparation, kind of manipulating the beetle. Then it does the same thing as in the last video, but it's not clear if it's host feeding or just breaking the membrane so the parasitoid larvae can feed through it, or to make it so that the gap stays open so it can lay an egg. And then it turns around, and then if you watch, it's going to draw its abdomen over the beetle, and you can see the egg being deposited in that same spot. Its usual host is Hypothemis eruditus, and this is it trying to go after different but related species of beetle. It turns out this beetle knows how to defend itself against the wasp. Watch, it tries to sting it, and then the beetle clamps down on the back end of the wasp. The wasp tries to extricate itself. This is in fast motion now, takes a few minutes. And then watch, the beetle's gonna release. And then the wasp can move around again. The beetle seemed unaffected by this stinging attempt and did its normal, normal beetle stuff for the next few days. These are Cephalonomia species. It turns out there are at least 2 cryptic species close to Cephalonomia hyalinopenis here. They seem to do something similar, which is like the prorops to parasitize Hypothemis eruditus, but these go after the larvae and pupae. Here's a wasp in a bark beetle tunnel, and you can see the bark beetle larva here with the small parasitoid larva at the bottom. This is the species we find in macadamia nut husks stinging a larva. This is a larva of the coffee berry borer, which seems to take an interest in. Cephalonomia hyalinopenis is known to parasitize the coffee berry borer, though we haven't found any of these similar species in coffee fields in Hawaii. And these seem to take much less interest when the larvae are in their natural environment in coffee berries. This is it either attempting to host feed or just taste the cuticle of a larva, now Hypothemis eruditus. Notice how it kind of moves its mandibles alternately like that. This behavior often kind of continues for some time, about 10 minutes before it goes on to do something else. Here are 2 more cryptic species in the genus Ecphyllus. One we found associated with a variety of trees, and the other we have found only on how trees going after Tylopodius beetles. On the right is what they do. This is a branch with the bark peeled off. On the top is the bark here, and on the bottom is the rest of the branch. They drill through the wood from the outside and oviposit into their hosts under the bark. The larvae develop as ectoparasitoids, and then they spin this pupal cocoon and then chew their way out through the pupal cocoon and then through the bark, making these small holes. Here's one ovipositing. It has to somehow find the larva under the wood so it can lay an egg in it. Many wasps do something like this, and though it's been studied, it's not totally clear in general how this happens. It does this routine, finding a promising location and tapping the wood with its antennae and turning, and then here it's going to give it a try to drill down. It didn't like that, and then it's gonna try again. There it goes. And this time was actually successful and parasitized the larva and somehow passes an egg through that ovipositor. This is also sped up. It takes about 1.5 minutes in real time.
Jessica Hyde
joined the meeting.
And then it's done, and it goes off to search for another one. These are the last ones we'll talk about. You can see in the top left picture what kind of looks like a drop of liquid coming out of the beetle larva. That's the belly of a mite, and the rest of it is very tiny and can't really see it in this picture. Sometimes a few of these are attached to the larvae and seem to result in the larva drying up. In this picture in the bottom right here, you can see this one. This one has a few that are hard to see, and this kind of round thing here is one mite, and the larvae is almost desiccated. So here we've only talked about just a small bit of the biodiversity that's out there flying around, and here are just a few others that we found emerging from wood. Most of them we don't know what they do. The one in the box in the middle there is Cerocephala ecoptogastri, which we've confirmed going after bark beetles. Thanks for listening, everybody. Thanks for being here.
Thanks, David. Awesome presentation. Very nice videos and everything. Lots of information. Thank you. So if anyone has questions for David, please type in questions in the chat box. For now, we will have another presentation from John Herati. John Herati. John, are you ready?
I'm ready.
Okay. So John is professor of entomology from Department of Entomology, University of California, Riverside. His research is focused on the systematics, phylogeny, and biogeography of Chalcidodi wasp. That means he travels around the world in different parts of the world. And he's also here on Big Island to study the ant parasitoids, especially for, I think, little fire ant parasitoid. And he will be talking about a new ant parasitoid on Big Island, what it is and how will it do. John.
Well, thank you, Roshan, for inviting me. And I wish you hadn't put me after such a wonderful speaker. David did a great job. So I'll talk to you a little about my exciting little ant parasitoids. So one of the things about this whole thing is it's a rather unusual story. I spent the last 45 years of my life or more working on these little parasitic wasps belonging to eucharidids, which are part of Chalcidoidea. They're all ant parasitoids. And I'll give you more information on that in a second. But all of this started as, and I swear to you, I do not live on Facebook, but I came across this one little posting here from Vail Rogers that he had this wasp that he had located in Hawaii. And I said, that's impossible. So I contacted him and made sure that that's exactly what he had found, because this is a eucharidid wasp, which doesn't occur in Hawaii until now. And this is where he collected it. He was able to identify me that he said just behind the USDA Forest Service Institute of the Pacific on the Big Island, right there, he ran a trap and he was able to collect these wasps. So I had a confirmation on this. Then I thankfully got a hold of Ellen Bitume that's working in the same facility, and Vail had already left. And she went out and ran a trap for me. And all of these are the ant parasitoids within that one little trap, along with their host, which is that Wasmannia oropunctata, that little red fire ant. Bill Pereira, who is here, had a wonderful background to this. He's been running a lot of yellow pan trap surveys around the island, and as well as sort of picking up, these collections in Hilo. He had a number of other localities where he had collected this, and this is what we were able to publish in the paper. And he's even got it now on the other side of the Big Island. So this thing is rapidly spread all around the island, in at least the lowland areas, since probably after 2019, when Bill started making these original finds. So just all of this came out in a recent paper with the Bishop Museum Occasional Papers. And I want to again thank a lot of the people on here that were helpful in gathering all of this information. So Vail Rogers, Tracy Johnson, Bill Pereira, and my grad student, Austin Baker, Ellen Batum, and a couple of my one previous grad student and one previous collaborator. Anyway, so this is what we know about Hawaii. This was actually a really weird thing, because back in 1994, I published a paper that said there were two possible biocontrol agents. One that went after the imported fire ant, the nasty one, Solenopsis, and the other one was after the little red fire ant, Wasmannia. So there were two candidates, of which I thought the one going after Wasmannia was the best opportunity, but there's lots of other things that stopped that from moving forward. But in part of this, trying to sort of understand what was going on with this one that was attacking Wasmannia, I had a project back in 2009 and 2010 to look at these things on the island of Dominica. So I got some background information that is helpful to understanding what exactly is going on with this wasp. The roads there are a little bit challenging sometimes. These people all helped me here. Brendan Morris, Elizabeth Murray, Laura Verone, a couple of undergraduates, Jessica Ortiz and Jasmine Soto, as well as the people that were helping out in Dominica. And we were able to go up there. And one of the things I wanted to bring into this is, I had a presentation on these wasps from Dominica. And also, we don't know anything of what's going on in Hawaii, except for little tidbits of information, but we do know a lot about Dominica. For those of you who have never been there, it's called Nature Island. So it's one of the most mountainous Caribbean islands, which makes it, very similar to some of Hawaii conditions on what these things are occurring from. It's got a wet season, it's got a small population, it's been an independent nation. We take that and we overlay that with what we know about Wasmannia oropunctata. Basically it's an invasive, it's one of the top 100 invasive species. It's widespread throughout South America. It's probably been introduced to the Caribbean, although there's questionable things about where it was there, how long it's been there. It's a human nuisance and it's a biodiversity destroyer. And I say that I have at least personal experience with that, where I've seen it in the Galapagos, where when it gets somewhere where there's extra floral nectaries available or things like that, it can dominate a landscape and just remove everything that's there of other kinds. So I think that's one of the worst things that I've been able to see on this. Anyway, everybody knows how bad this thing is and how invasive it is, and especially in island situations. So what are these eucaryotid wasps that I'm talking about? Here we have the genus Orosema. I'm only going to talk about this one genus Orosema because that's the only one that's relevant. There's lots of other weird things that go in the family. But these ones in particular, they're always parasitoids of Myrmecinae, which include our nasty ants like Phaedole, Solenopsis, and Wasmannia. They always insert their eggs into leaves, sometimes into the bracts of flower buds or along stems or things like that. They have even been known to oviposit into bananas. And then back in the 1950s, they were a major threat to bananas in Costa Rica, and they had the United Fruit Company had a big program to try and figure out how to control or eliminate what these were. Sometimes they use an intermediate host to get into their host. Sometimes they don't. They always attack ant larvae. They're internal in the first instar on that ant larva. They develop on the ant pupa. The ants love them. They carry them around. They protect them. They treat them in all sorts of nice ways. And because they're myrmecine ants, they have no cocoon, so they're just out open in the brood pile like that. Basically, similar biology you're going to get between any of the Wasmannia parasites or some of these other ones. This Orosema minutissima is found across northern South America. It's commonly collected in the Caribbean. It's been reared from Wasmannia oropunctata, so we have definitive records from Dominica and Puerto Rico. We have it reared from Phaedole in Dominica, proposed as a biocontrol agent back in my 94 paper. It does oviposit into the leaf surface of a variety of plants, and I'll show you those examples. We get one other, in Dominica, we get one other rare species, which is this near costaricensis that, I'll give you a bit of information on that in a minute. But as far as biocontrol issues, what do we care about? Host plant specificity, establishment, potential pest status. Are these things going to be causing damage on plants that people care about? Host ant specificity. What are they going to be attacking? If I was importing this to a mainland situation, it would be bad because there's other myrmecine ants. As far as I know, basically all of the island locations where Wasmannia has taken off, there are no other myrmecine ants. So there's nothing else that it can actually attack in terms of natives. Orosema costaricensis is known to oviposit in the skin of bananas. It's an old thing. I'll show you some things that I looked up on that. And then again, is it actually lowering the actual populations of Wasmannia? So is it actually worth any effort? The nice thing about Hawaii now is it's been accidentally introduced. So we don't have to go through all the importation and all the deals with quarantine to be able to look at this and see whether it's actually doing something. Just some things to show you that Dominica is much like many of the locations in Hawaii. This is your old Google Maps thing. It's got various locations where you can find wet forest, somewhat dry forest. This one location here is actually where that other species of Orosema was collected. It's a drier habitat than most, but it is something off there. These are some just other, pretty locations. In Dominica, it was a great place to spend a couple of trips. You will not find either Wasmannia or the parasitoids on the beach. You won't find them on the beachy rocks. You won't find them in the pools, but you will find them in these nice little habitats over like that. Of the sites I looked at in bananas in Dominica, here's where I know whether these Wasmannia are all over the place. I never saw any marks on any of the fruit. So there doesn't seem to be that, it's not something that they've ever worried about, they've ever seen. So whatever happened back in the 1950s in Costa Rica seems to have been an accident. It may be associated with different species of Orosema, but it also might be associated with pesticide upsets at that time. So I don't think it's never been recorded as a pest of a fruit of any kind. When we were in Dominica, we were trying various ways of trying to collect these by using like Malaise traps to pick up various things. So I'm more interested in all general insects. I had some wonderful helpers to go around and use fine mesh insect sweep nets so we could do general surveys to find out where we were finding these things. We're using, straight tube aspirators, and we're collecting these into vials for later sorting. Sometimes we get a little bit too much work and people get really sort of, upset, all the extra labor they have to go to look at all these specimens. What we did find, which is really unusual, is that these wasps in particular, the whole genus Orosema, of all the species that are in it, and there's probably, a couple of hundred, this one species loves to go into yellow pan traps. And if you don't know what a yellow pan trap is, it's just a little yellow plastic bowl with some water and a couple of drops of soap in that. And you basically go along, you use a fish net, and you can strain these things through and sort them back in the lab and things like that. Again, you can sort of see conditions on Hawaii might be very matched to Dominica in some cases when it gets very hot and sweaty. This is a blow up of that pan trap over here to show you how it operates. We have another different trap type over here that we've been using. So within this, you can see that one little thing there, and that's one of the wasps that's actually committed suicide into that fluid with the soap in it. It's an easy way to sample these things and find them. This is where that Ellen was able to find them behind the forestry lab in Hilo, just by setting out some of these pan traps and collecting these up. My way of sampling for Wasmannia is different than most. It's a white card with a little bit of peanut butter in the middle, and I can assess sort of population densities here to sort of get an idea of what's happening on the ants in the area. So I can pair these things up for survey purposes. Of course, you get some other insects coming in there and feeding on the peanut butter, and every time Dominica, you also get some land crabs coming in and feeding on the peanut butter, and so it's a little bit disruptive, but generally it works. Okay, so what did we find? The cool thing is about this one is there's actually 3 discrete size classes. So we find Orosema minutissima basically all over the island, which is sort of similar to what we're starting to find in Hilo. We don't have any elevation similar where we can sort of look at elevational gradients, but generally across the island, they're pretty much everywhere. These small ones are always associated with Wasmannia. The 2 larger sizes, we found that we've been able to document that are actually associated with Phaedoli on the islands, and we've actually got rearing records of that. These undergrads that I had in my lab started off with a sequencing project, and we started looking at Orosema minutissima, these small, medium, and large sizes over here, and they come up with identical sequence for whether, 3 different gene regions. So it seems like everything in the Caribbean is potentially recent and potentially exactly the same haplotype within these things. And these are both, Phaedoli and Wasmannia parasitoids. This was our original map. Again, the interesting thing here for those who know that Wasmannia oropunctata in Hawaii, they say that potentially one of the original distributions or importations came from Florida. There are no records of Wasmannia, of this Orosema minutissima from Florida at all. This is a different species of Orosema, which has now been called Evansii. So now we have this distribution mostly confined down through here. Now we published on this in 2018, same results with showing that different countries all through the Caribbean and different sizes all came up with identical haplotypes in here. So we have a general distribution of the small haplotype, and then we have this medium to large haplotype over here. And I mentioned that extra little Orosema I collected off in the dry region in Dominica. That's this thing over here, which is closely related to what we see as Orosema costarricensis, but something out in its own long branch here. But anyway, this is what we're concentrating on down here, this minutissima. We look at this typical biology, these larvae, they have a weird first instar larva that goes off in search of its host. I'll show you that in a second. Once they get into the nest, they actually attach to the ant larva, and here are a couple developing on here. They become internal parasitoids of that first instar. When the ant pupates, they come out to the external surface. They start developing on it, and you see those little lines there. That's all the first instar. It just starts swelling up in enormous size. Continues its development, then basically sets up an intermediate prepupal stage larva, then it pupates, and then you can find it in the nest like that as the pupa. So they're really easy to spot once you crack open a nest. These are the ideas of the plant host damage that you can get from these things. And so this is on a fern here, the alien Soren fern. These are the things, the records in Hawaii. There's basically a similar host range in Dominica. So they'll either go after ferns or they'll go after broadleaf plants as well. And here we have little egg punctures over here. You can see these are old egg punctures here where the wasp has got a big ovipositor and it slips it into the plant and lays a single egg with each one of these oviposition punctures going along through in here. So it'll scatter on the leaf. The thing is interesting is it's scarification of new growth, but when you look at the old growth, it just sort of disappears. It's not a long-lasting impact on the plant. But when you get high densities, you can get the plants covered with all these little dots on them. And over here, if you want to look what comes out of those little dots, you got to focus on that little spot right there. And this is Ellen's video of one from Hawaii. And that's the larva, how it's hatched, and it's going out and it's crawling, and it's basically going to be getting in touch with the adult ant and then being carried back into the ant nest where it's going to be attacking the ant larva. So, and then here's more of those punctures up in here. This is a broadleaf plant. You can see the little marks and puncture scars that are over on here. This is some work where we looked at different sort of where were they laying their eggs. We were interested in trying to find out some different things. So whether they were laying on dorsal fern leaves, that seemed to be the preference. Was most eggs were laid on the dorsal surface. On the ventral surface, we get just a few eggs. Dorsal broadleaf, 11.5, dorsal fern, 145, dorsal broadleaf, 38, and ventral broadleaf, 7.8. So they seem to like laying them on the upper surface. I don't know why. What's interesting to me, though, in particular as well, which isn't shown by this, is this area here, where Elizabeth and Laura are sampling. This is where we found Orosema minutissima ovipositing onto the ferns, and this is where we get lots of the Wasmannia. Further up the path, there's an area of almost dominated by short broadleaf ferns, and this is where we almost always found the Phaedoli larger size classes of Phaedoli associated large classes on those. So there's a behavioral difference here between what they're going after in different areas that we don't really understand. Okay, just some little things about looking at the number of pans. This to me was just astounding that these are just pans, 20 pans and bait stations. We put them down for an hour. At this one place in T2 Gorge, we were able in 1 hour to collect over 35 Orosema minutissima and in quite a density of the plant pans. So it's really an easy way to go out and sample these things. And we can look at this one where we can compare 20 bait stations over 5 hours, each one of these being run for an hour. And again, we're getting similar numbers, high numbers of minutissima. So they're all over the place and just, again, jumping into these pan traps. That makes an easy sampling routine. So look at the ants that are dominating the bait stations in those different areas. We get Wasmannia oropunctata is mostly there. We get Phaedoli in the other aspects, and then some other trailing different ants that keep coming in there. These are the actual reared ones. So this is the larger one, which is associated with two different, you might imagine that the two large classes, medium and large, are going to be associated with the workers or the soldiers of the Phaedoli. And these are the Wasmannia associated one, where that's the Orosema pupa, and that's the pupa of the Wasmannia down in here. So back to Hawaii. What's interesting is the orange are basically the three different size classes we get in Dominica and throughout the Caribbean, where we get basically the Wasmannia associated species here, and then we get the Phaedoli assumed associates here. When we measured the Hawaii specimens, we ended up with our Wasmannia associates as we expected, but we also, thanks to Bill's search efforts, we actually managed to find a couple of larger forms up over in here, which aren't as large as our Phaedoli associates. And one of the questions we have is, are these attacking the Phaedoli in Hawaii, which is why we're getting that large size, or potentially are they attacking reproductives of Wasmannia, which might result to them also coming out with large size? These, again, are things that we're unknown that we still need to figure out. So as far as everybody here is concerned, is biocontrol, will it work? It's difficult to assess the impact on Wasmannia because it's a numbers game. For one thing, I have not been in Hawaii and seen how bad it is. From some of the reports you tell me about how bad Wasmannia is, it sounds like it's really bad. Where I've been in Dominica and Costa Rica, I've never seen Wasmannia that bad. I have seen it really bad in the Galapagos. So it's sort of getting an idea of just what the numbers are. But also this, when I talk about a numbers game, trying to get parasitism rates on wild collections of Wasmannia brood is something that I don't consider to be an easy problem. And I'm a systematist, not an ecologist. I want to emphasize that. So all of my sort of ecological experiments are pretty rudimentary. We've observed Wasmannia in Costa Rica, Dominica, and Trinidad. Again, it doesn't seem to be a big problem there. I'd love to get a better comparison. I have not been to Hawaii, by the way, to sample this thing yet. So this is all just third-hand information at the moment. It's hard to assess the impact of a wasp when they're everywhere. This is the problem I always faced in the Caribbean was, if every island has both the wasp and the ant, how do you assess whether one is actually having an impact or not? You don't have anything that you can look at a controlled experiment, which down in my last point here, down here is, there is that possibility in the Hawaiian Islands, because now we have the big island that has the wasp as well as the ant. And we do have some other islands where we do not have the ant possible. So it's going to be possible, you know, if I get somebody interested, it's a good way that we might be able to assess actual impact of this thing. Right now it's found widespread in the lowlands of Hawaii. We really need to go over and get a survey to find out as soon as possible what these are. And you know, I still think pan traps are best. I'd like my peanut butter bait cards, but I'm hoping to use something which is a little bit more updated, where we could lay these things out, do a QR code, just do a photographic assay of these things, where we could get georeferenced images, and we could come through and get an idea of what is our ant densities out there, and then using those yellow pan traps, find out just what is our occurrence of those different wasps. So I have lots of people to thank for all this. Don't have any images of the people in Hawaii that have helped with all this project, but you know, it's basically been a lot of work by a lot of people to get some general understanding. And I wanted to end off with, I have this little life cycle of an ant parasitoid pamphlet. And if anybody just wants to email me, I can send you a PDF of this, and you can post it or spread it around or do whatever you want. It's focused on a different species of Orosema in this thing, but it's basically essentially the same general biology that's on there. And with that, I will say thanks.
Thank you, John. That's a very good information. So one thing I have a little bit confused. So these ant parasitoids also lays oviposit on the plant tissue, different plant tissue. Is that right?
Right. So the oviposit, so if you look at this little life cycle diagram here, so they're ovipositing onto the plant over here. These ones are associated with, some of them are associated with extrafloral nectaries. Some of them seem to be associated with flowers, and they jump onto thrips. And the ants are picking these things up. Ants don't eat solid food. So what they do is they're actually picking them up in their mouth parts. So they pick up solid food in a pouch in their mouth parts, and they use that, and they eventually feed the ant larvae with that solid food. And they usually, you know, take nourishment from the ants or from the ant larvae as they regurgitate the liquids. So getting into the nest.
So that means these parasitoids feed on the ant larvae, right?
They feed on the ant, well, they attack the ant larva down in here, but they develop on the ant pupa. So they're always going to the same portion on the ant pupa, and they're always developing on the ant pupa. So they're very specific that way. How they actually get into the nest here is always a big challenge to uncover, because across the entire family, they use all sorts of different behaviors. When I see like that question of why do Orosema minutissima oviposit on the tops of the leaves, and I've seen in Dominica, Wasmannia will, I don't know if I can show, make sure I can see myself there. In Dominica, though, the Wasmannia will go across the top of a fern leaf, and its head is going back and forth as it walks and is looking for foraging items. And I think what it's doing is it's finding these planidia, picking them up in its mouth parts, and then bringing it back as food to the ant nest directly.
So again, one more question. So these ants will feed on the larva, then they will oviposit on pupae. Is that right?
No, everything by the wasp is done outside of the nest. So they have to leave the nest at this point, get out of the nest. They mate out here. They oviposit in the plant tissue. It's that small little first instar larva, that one that I showed the video of, that tiny little larva. That's the one that now has to get into the nest, get onto the ant larva. And it will sit on that ant larva, and it waits until the ant pupates. And then when it pupates, then that's when it starts developing on the ant pupa. When I say about the complexity of measuring parasitism, the one thing I noticed in Dominica, which I haven't seen, is I think Wasmannia actually partitions the brood in different caches. So they'll keep all their larvae together and they'll keep all their pupae together. So when you're trying to track down what a nest is, they can be spread all through the vegetation and in different areas and under leaves and things like that, it's hard to say, okay, what is the whole colony? What is the whole colony parasitism going on? But I think right now in Hawaii, you got a perfect opportunity to be able to sit there and say, okay, so what's its impact on the Big Island? And then if you look at the other islands where Wasmannia is in similar habitats but not attacked, what's it like? And maybe eventually you could even move the parasitoids to those islands and see if it has an actual impact on these things. And I mean, it's important. It's an important wasp to me because I think it's got great potential for the Galapagos and for basically almost any island situation in the Pacific where myrmecines are not a native ant species that you have to worry about. These are a perfect thing. The reason I've never suggested them for a biocontrol agent is I can't imagine how you can rear them and test them for host specificity. So by accidentally getting Hawaii, you got a bonus.
So we have this parasitoid on Big Island.
Yes.
Right? Okay. So do we know the impact of this parasitoid on the little fire ant here on Big Island?
No.
No.
That's what I actually have some money to come over and I want to try and do some initial studies and see if I can actually, see what that's like. But I'm really looking for someone in Hawaii to actually think about trying to pick this up as a research program because I think it's a great one.
Yeah, sure. You're welcome. Yeah, I'm here on Kauai. Kauai has also recently detected little fire ant in one location. So yeah, there is a scope. Yeah. Yeah. Thank you.
And I think this whole idea of yellow pan traps and things like that, I mean, this is something which is easy to do. And I would encourage anybody to run out and grab some yellow pan traps and just take a look and see if, especially if you can correlate it with finding, using some peanut butter to look for Wasmannia. And I would say anywhere in the Big Island now, if you have ferns in the lowland areas and you have Wasmannia, I'm willing to bet you have the parasitoids.
Okay, questions for David and John. David, so I have a question.
Yes.
So you talked about many different kinds of parasitic wasps. Yeah, some are larval parasitoid, some are pupae, and some feeds on the adult in between the prothorax and mesothorax, and like a different kinds of parasites. Can you a little bit generalize this?
So I guess worldwide, most parasitoids of bark beetles go after the larval or pupal stage just because they're softer and they don't move or defend themselves very much. It makes it I guess more likely to evolve. And many of them do it from the outside. They go in through the surface of the wood to get the ones that are living in that phloem later. So that's probably the most common way parasitoids go about getting bark beetles. So it's less. So some of those ones at videos of they go actually into the wood, right? And so it's kind of hard to say whether that's less common of a strategy or it's just less seen because you just have to be breaking open wood and that's often a little bit more difficult to get at. So one thing I didn't mention is about the shot hole borers. Somebody's been looking for at Riverside. They've been looking at parasitoids of them, and it seems like they found some important ones so those are ones that actually go into the zyome layer. So I don't know if you look for it, they're probably there is my guess. More than people know.
Okay, so again, one more question. They oviposit through the wood, or they go inside the hole, bore hole and find the larvae or pupae?
So both. There are wasps that do both, but the more, at least to human knowledge, more common strategy is to oviposit through the wood from the outside. But yeah, there are ones that you can call them cryptoparasitic that go inside the wood.
Okay. So there is one question asking what type of bark beetle or boring beetle affects avocado? If you know.
Well, there are a
Okay.
few, but the kind of important ones is that shot-hole borer, because it can spread so there's two species of shot-hole borers here. There's Uolacea fornicatus and Uolacea probrevis. The fornicatus, they have different fungal symbionts, and the fornicatus can actually spread its Fusarium symbiont to avocado trees, and it can kill the trees, which is another thing is that one tends to go into somewhat living wood. So you can have a somewhat healthy tree, or maybe a branch that the avocado tree is not so happy with and isn't paying that much attention to, and maybe they'll go in there and they could they can kill the trees. So that's an issue.
Okay, thank you. So it says David wins the best horror movie ever presented. Kudos for filmmaking. Thank you. Okay. So there's a question asking how many beetles does a single wasp parasitize?
I think it's pretty variable, and I don't really know the answer in general. That Phimasticus, so if you squish them, you can see the eggs that are inside them, and they only live for 3 days, so they probably have produced in some stage all their eggs before during their development. So I think there's been about 25 eggs in those, just roughly, and like 20 to 30 typically. And so each one typically lays 2 eggs in a host. So divide that by 2. And so maybe they, if they can get rid of all their eggs, they're going after 10, 15 or so. In general, though, that's, and some live for a long time and keep producing eggs and just keep doing it as long as they can, I guess.
So you said parasitoid will lay many eggs, and
So,
maybe probably 2 eggs per each individual larvae or pupae or the adult, right?
So the 2 egg thing, that's just the Phimasticus. It kind of has a special biology. There's just a lot of variability. Some will lay 1 egg on a host. Some will lay lots of eggs on a host, like Bacillus. They sometimes do that. And then
if they lay lots of egg in the host, lots of parasitoid will come out.
Yeah. It looks like you have a larva and then a lot of little, sausages coming out of it, and then they'll all pupate and turn into adults.
So another question also relates similarly. It says, do these parasitoids always work alone? Have you seen them work in pairs or group?
So
different things.
About the Bacillus again, there's some that actually do work in groups. Most, I'd say, most are pretty solitary. Some of those Bacillus, they go after cerambicids or moth larvae sometimes, and they have, especially the cerambicids, they have kind of big mandibles, and so they can bite the wasps, and so it's kind of a little bit risky for the wasp, and they actually get together. A few individuals come, and there's some Goniosis parasitoids that do this, and they will sting the larvae and subdue it together. And then they also kind of sometimes take care of their brood too. The females who have oviposited stick around and try to help out, I guess.
Yeah, sure. Yeah. There's another question for David. Is there any interaction between wood fungus, bark beetle, and the beetle parasitoid that you might observe?
I don't know. Yeah. So the beetles often carry a fungus that they use to help them feed on the wood that will digest the cellulose and things like that. And it's likely that some of the specificity that these parasitoids have is mediated maybe by the fungal symbiont, but don't know. That's just a guess. And that's actually something that I'm trying to figure out.
Okay, thank you. So another question, it's for Beetle, again, Beetle, yeah, for David. What is the plausibility of wasps moving through a beetle population and picking up and spreading pathogens from beetle galleries?
That's a really good question. Yeah, that's a nice thought.
Mhm.
I don't know. I guess, yeah, good question. Things can, attach, and in addition to um, pathogens, you could maybe even think about mites that are phoretic and, hitch a ride on wasps or something and travel between beetle galleries. Yeah, it's a great question.
Yes. Yeah. So another question, this might be a question for both of you. Are you aware of any other parasitic wasp in Hawaii that sting humans? I think,
No,
okay, so there is
So,
some answer also.
I mean, there's the big wasp. There's the big wasp you always have to
Mhm.
worry about. But as far as the parasitic wasp,
Okay.
they're the most wonderful little creatures. They don't attack humans. They're only worried about their business.
Okay.
yeah, I saw in the
Yeah.
the chat that
Yes.
came up, and so somebody brought up Cephalonomia gallicola, which is, and I think Carl gave a pretty good answer in there already, but
Yes.
they're typically, I think they go after anoboeids that are pests of, like grains and stuff, so they can often be found in houses.
Mhm.
And so you're just more likely to interact with them. And so maybe somebody gets, these bacillus, they might sting, but most parasitoid wasps are minuscule. They just have no interest or cannot sting humans. It's so there's also Sclerodermis immigrans, which is possibly will sting, but I've had them in my hands plenty, and they. I think I got a little prick once, but it's very unlikely that you even come into contact with a lot of these things that are not associated with houses, and there aren't very many that are associated with houses.
Yeah, they're definitely not aggressive stingers, so they'll sting you if they like get under your clothes and then you sort of pinch them. And that's one of the things with Sclerodermis immigrans is it attacks like wood boring beetles that can be in furniture, and so that's the way that people typically come into contact with them. And then the Cephalonomia gallicola attacks like flower beetles or drugstore beetles, so they can be in pantries and stuff. And that's how I've gotten stung by them out in the forest.
Mhm.
I've had them drop down the back of my shirt and get caught in my collar and then sting the back of my neck.
Thank you, Carl, for your answer and other notes in the chat box and as well as in person. So I will just read two questions because we are a little bit lagging behind. So I think this question is for John. Are there any studies showing lower Wasmannia density in ant traps in the areas with Oracema compared with Wasmannia density in the areas without Oracema?
No, I haven't seen any. I have not seen any. I mean, that's the problem. Is every area that in the Caribbean, everywhere you go, you get both the ants and the wasps, pretty much. So it's hard to and like I said, I don't see a problem there that, I don't know exactly how bad it is, but the way people describe it to me in Hawaii, it can be a pretty nasty experience to get it into a Wasmannia infested area, which is not what you see in the Caribbean. They tend to be just, they're there, but they're not obnoxious. And we've been trying to do these density samples to look at on peanut butter bait cards and things like that to find out if we can do it. But realistically, what I need is a controlled sort of situation where I can have a large area where there's only Wasmannia and there's a large area where there's a Wasmannia with the parasitoids and actually see if that's actually working. And so Hawaii is the perfect experimental arena for this.
Thank you, John. So this will be the last question we discuss. Then can you both answer question in the chat box? Right. So this is the last question. why do the ants take the parasitoid larvae back to its nest?
Sorry, what was that?
Why do the ants take the parasitoid larvae back to its nest?
Because well, I guess it's actually, well, they think it's food, I think it's all about food. It's association with a food resource and across the entire family. I think the more and more cases I get it broken down, it's about the ants are going out foraging, and basically they're picking up these little particles, which, you know, they're 1/10 of a millimeter in size. I've got images where I've got big ants with their mouth open and they've got planidia, like the larvae are sitting inside their mouth parts in there. So they don't notice it. And then they're just going back and feeding it to the ant larvae. And sometimes the ant larvae will have most of the larvae attached around their neck region, which is a good idea that that's where the ant was coming down. And these things are alive within the mouth parts of the ant. I've seen that. So it's just a case of, you know, you're fooling the enemy by coming in on the, you know, these tiny little larvae that they just can't notice. And there may be other ways that I don't know, because there's weird ones, because I have one in California that's feeding on desert willow. My hypothesis was that the Phaedole was coming up and feeding at the nectaries, because in that the larvae, they concentrate into the nectaries of the plant. So it seems to be an easy case that the ants are coming along and picking up the larvae with the nectar. Except that we've dissected thousands of the adult ants that are foraging, and we've never found anything in their mouth parts. But if we dissect Camponotus and we dissect Myrmecocystis, some other large frugivorous species, we get lots of planidia inside. We get 20% of the adults have these planidia in their mouth parts. And then some undergrads of mine found some dead ants of Camponotus in a nest of Phaedole desertorum. So we actually now have some ideas that the Phaedole may actually be scavenging the dead Camponotus and then using that as bringing that into the ant nest. So there's mystery stories all over the place that still need to be resolved.
Okay, thank you, David. Thank you, John. I can see 10 more new messages, so we need to go move along. And if you both can answer a question in the chat box, will be great. And I will moving on to next presentation. Kyle, are you around? Hello.
Hello. Hey, Roshan. How are you
Hey,
doing?
good. So our next presentation is from Kylie Roy. She's a geneticist from U.S. Geological Survey based on volcano Big Island. And even she's a geneticist. She does entomological work. And she's one key member of a project studying the role of ambrosia beetle in spread of rapid ohia death in Hawaii. And I think she recently graduated from Purdue University. She completed PhD from Purdue University on chemical ecology of ambrosia beetles, right?
I did not graduate.
Oh, okay. Still continuing.
But I appreciate
Okay.
the compliment. But
Okay.
yeah, I'm a second year PhD student at Purdue University. Yeah.
Okay, sorry. So we will be talking about ambrosia beetles and rapid ohia death and so on. So Kylie, whenever you are ready.
So thank you for the introduction, Roshan. So I'll be talking about the role of ambrosia beetles in rapid ohia death. And so this is really focusing on how we've been proving that beetles are involved with the spread of ROD. And then at the end, I'll touch on some current management strategies. And so just to give you guys a brief introduction to rapid ohia death. Hopefully a lot of you guys know about it. And so it's actually a fungal disease caused by two different fungi, Ceratocystis leucohea and Ceratocystis huliohea. And leucohea is much more virulent than huliohea, so it's known as Ceratocystis wilt. And Ceratocystis huliohea has been given this name of ohia turnover, so that huli, and so it causes more of a canker. So both of the fungi work to clog the transport of water in the sapwood, so the functional part of the xylem. And in order for the disease to spread, there needs to be some sort of wound or ingression. And so the host is Metrosideros polymorpha. They're in the myrtle family. Their Hawaiian name is Ohia lehua, and we call it Ohia for short. It is arguably the most ecologically and culturally important tree in Hawaii. So I think a lot of people have been talking about ambrosia beetles, even last session with Mark Hughes, so I can be super brief on this. So they live in the xylem of trees, also where Ceratocystis lives, and they are fungus farmers. So they actually plant and cultivate their own ambrosia fungi. And in some systems, the fungi that they're planting is actually really pathogenic, such as in laurel wilt. But fortunately with Ceratocystis, their ambrosia fungi is not what's killing the trees. So it's not Ceratocystis. And we're specifically talking about beetles in the tribe Xylobarini. And so these beetles are known to be invasive because they have this really effective invasive species, because they have this haploid diploid biology where a single unfertilized female can potentially start an entire population in a new location. And so if this habitat is suitable, they can actually remain in that gallery for multiple generations. And we've also seen that happen with Rapidohea death. And these beetles are generally attracted to stressed and dying trees. So anything that will disturb aerobic respiration in the trees will cause a breakdown in the respiration, and this ethanol scent will come out and attract the beetles. So anything that will stress the beetles, stress the trees, such as drought, nutrient limitation, flooding, fire, or wounding, will cause this ethanol scent and bring in beetles. So this talk really focuses on the indirect transmission and the direct transmission. And so briefly, indirect transmission refers to the movement of frass that the beetles are creating when they're making their galleries. There can be viable Ceratocystis propagules that is pushed out into the environment. And then this viable inoculum could then get moved in the soil, moved around in pots, moved on your boots or in vehicles, also blown in the wind, and even potentially spread via waterways. And so direct transmission refers to the beetles actually moving sticky spores. And let me just show you here. So the sexual form of Ceratocystis produces this long perithecial necks and these sticky ascospore masses. And to show you just how sticky they are, these are a bunch of beetles in a culture of Ceratocystis, and you can see all these ascospore masses stuck on their bodies. And we have found 5 ambrosia beetle species on Hawaii Island that are commonly associated with rod. And I just wanted to mention that most of them are invasive species or introduced, and they've been associated with other diseases as well. There's more things you can read about in this paper if you're interested. And a quick update on work that I've been doing with Roshan on Kauai. So we've found all these same ambrosia beetle species associated with rod on Kauai, but except for, of course, the Hawaii Island endemic Xyloborus similimus. And we have also found another beetle species, a platypodid beetle in the genus Chrysopteris on Kauai as well. And so now really breaking into the proving the indirect transmission and the direct transmission of ambrosia beetles in the rod pathosystem. And so there's these things called Koch's postulates, where there's 4 criteria designed to establish a causative relationship between a microbe and a disease. So we had to go about these things to be able to prove that frass actually acts as a transport mechanism or a sort of vector. And so first you have to prove that the microorganism is found in diseased plants, but not healthy plants. And then you have to be able to culture the microorganism from the diseased individual, then take that and re-inoculate it into a healthy individual, and then re-isolate the microorganism from the inoculated individual. And so this is a paper that we are about to submit to the Journal of Plant Pathology, and the lead author is Dr. Mark Hughes at the Forest Service. And so this project looked at 4 different aspects of indirect transmission. And the first was really to understand how often does frass from ohia contain ceratocystis. The second was completing Cook's postulates. Can frass directly kill ohia seedlings? The third part was trying to understand the spore type that we often find in frass. And finally, we wanted to know how long frass can survive, how long ceratocystis can survive in frass. And so we found, just based on a survey on Hawaii Island, frass is pretty likely to contain ceratocystis DNA. So 79% of those samples contain ceratocystis DNA. And 48% of them were ceratocystis leucohea and 25% were ceratocystis huliohea. And we also found 6% of the frass samples were co-infected with both ceratocystis fungi. And surprisingly, 72% of those frass samples also contained viable propagules. So the viable propagules can infect another tree. And in the second part of the study, where we're really completing Cook's postulates, and we wanted to know if frass can directly kill ohia seedlings, we conducted 2 different trials where we took frass and then inoculated seedlings with it. So you can see pictured here. And in the first trial, 40% of the ohia seedlings that were inoculated with ceratocystis were infected with the fungus, and those same seedlings also exhibited outward wilt symptoms. And in the second trial, 23% of the seedlings were infected with ceratocystis leucohea, and 15% of them wilted. So from these data, we really see that viability is variable, and it really depends on the spore load and other factors such as the age of the frass and other abiotic and biotic conditions. So the third part was understanding the spore type that is present in frass. And so we found these round spores, which are called chlamydospores. And there was no other type of spore present in frass. And chlamydospores are a type of resting spore, and it was kind of unusual because we were expecting to find Conidia. And so Conidia are darker pigmented, usually larger and probably longer resting spores than chlamydospores. And they've been regularly observed in plant cells and also in culture. And so what's interesting about this is that the chlamydospores appear to sort of be derived from Conidia, and the chlamydospores are less pigmented and potentially more easily activated than the resting Conidia. And we found that these spores, the number of spores in frass was extremely variable, although in general, you could find about an average of 1 chlamydospore in a frass particle. And the number of spores generally correlated with viability. And for the final part of the indirect transmission manuscript, we estimated the survival of ceratocystis leucohea in ambrosia beetle frass. And so how we did this was we collected bulk frass and placed them in different environments and collected subsamples over time to test for viability. And so we had 3 study sites, including a control incubator. And so these are the different markings so I can walk you guys through this. So we had a control incubator and the temperature and relative humidity here. And so the control incubator was set at 25 degrees Celsius, but it did not control for humidity, as you can see here. And the second site was in Hilo, which had a similar temperature to the incubator, but had a much higher humidity. And then the final site was in Volcano. And unfortunately, the data loggers died, but we can still get a good idea of what was happening. So it has a much lower temperature and also a very high humidity. And so in this graph, we're looking at the viability of the Stratocystis sample, so a total percentage over time. And as you can see, the control, which had basically no humidity, dropped off in viability right away. So at 8 weeks, and then it was no longer viable. And the Hilo samples, they kind of dropped off at the same time, but remained viable at a low percentage through 22 weeks. And then in Volcano, where it was there's cooler temperatures and higher humidity, we see that viability was, above, generally above 75% for the entire duration of the study, this whole 26-week study. So it could have potentially lasted longer, but we just ran out of frass. And so now transitioning into direct transmission by the ambrosia beetles. So in addition to Koch's postulates, in order to confirm an insect as a vector, you have to also follow Leach's rules. And so there's 4 general rules that we had to complete. The first is the insect has to be closely associated with diseased plants. And you have to be able to show that the insects make regular visits to healthy plants. And you also have to be able to prove that the pathogen is regularly associated with the insect. And then finally, you have to be able to show that visitation of healthy plants by pathogen-infested insects results in the disease. And so this is a manuscript that we'll be submitting to Forest Pathology. We're still collecting some data for this, so it'll be a little bit slower than the FRAS manuscript. And so this contained 3 different projects. And the first one was just understanding if the ambrosia beetles associated with rod actually carry Ceratocystis on their bodies. The second project was looking if beetles could directly transmit the fungi. And we looked at this through challenge experiments, forcing the beetles to attack seedlings. And the final part of the project is a wounding field trial. So we wanted to know if we could find ambrosia beetles regularly visiting wounded and unwounded healthy ohia. And so now we're looking at the first part of the project, the isolation of Ceratocystis from beetles. And so we conducted what we call aeration assays. So it's really just using an aquarium pump, water, and carrots used to culture Ceratocystis off the bodies of beetles, which you can see pictured here. And so we collected the beetles 3 different ways and subjected them to the different aerations. And so we collected beetles by rearing them from bolts in the lab, also from sticky cards that were affixed to ohia in the field. And finally, we collected beetles through baited traps in the field as well. And so you can see generally, so this is the percentage of beetles carrying rod fungi. Across the board through all 3 different collection methods, about 2.6% of all the beetles tested had viable propagules. But when you looked at individual species, you can see Xyloborus affinis really stuck out as more commonly being more likely to have Ceratocystis propagules on its body. And so this slide is now describing the beetle challenge experiments. And we're still in the process of conducting these. And so we have so far forced beetles to attack Xyloborhinus saxonii, Xyloborus affinis, and Xyloborus ferruginus. And we're working on Xyloborus perforans right now. And so what we did was we allowed the beetles to crawl on Ceratocystis leucohea and Ceratocystis huliohea, and then forced them to attack healthy seedlings. And you can see generally there was a high probability of seedling inoculation. And the final part is the direct transmission. So this is our wounding field trial. And so what we did was we monitored 5 unwounded healthy ohia and wounded 5 ohia in 2 different trials. So then we had a total of 10 wounded and 10 unwounded trees. So we made these wounds that were 4 by 4 centimeters onto healthy trees, and also tested them and made sure that they didn't actually have rot and they just weren't expressing the wilt symptoms. And we affixed a sticky card, as you can see pictured here, and monitored beetle attacks for 4 weeks. So we're going to be going back to check for wilt symptoms next month, because it will have been 6 months since we started the study. And so far, we've found 35 beetles on the wounded trees, 27 of which were Xyloborhinus saxonii and 3 were Xyloborus affinis. And on the unwounded trees, we've found a total of 6 beetles, 1 which was Xyloborhinus saxonii and 1 which was Xyloborus ferrugineus. So in conclusion, regarding indirect transmission and direct transmission, we completed Koch's postulates for indirect transmission and found that ambrosia beetle frass is an important vector of rod. And we were able to show that frass is commonly viable and it can also kill healthy individuals. We found that there are resting chlamydospores within frass particles that can survive for at least 6 months in cool and humid conditions. And in the direct transmission studies, we completed both Koch's postulates and Leach's rules, and have found that ambrosia beetles are what we call casual vectors of rod. And if we think about like the biology of things, beetles have the ability to actually ingress or make wounds into the trees, which is required for ceratocystis to spread, which may be more advantageous and require less chance than wind dispersal. Ceratocystis fungi are also adapted to attract insect vectors with their sticky sexual spores, and they also produce fruity smells, which is part of my PhD work, which I'm not talking about today. We hypothesize that direct transmission by ambrosia beetles are likely to occur in places where there are more invasive species and more alternate hosts for the ambrosia beetle species. So the trees are stressed, and which would cause them to attack the trees. So now just like really briefly, some current management. We've had, other projects going on regarding management. And so felling and tarping is still the best way to reduce the spread of rod. We have found that tarping reduces viability in bolts and also reduces beetle reproduction. So there's 2 wins there. And we've also been working on different semiochemical stuff. And so that's looking into the use of attractants and repellents. In trap trials, we've found that splat beetle repel and splat verb are both effective repellents. And we're hoping to obtain funding to work on applying these repellents to standing trees in the field. So I just want to acknowledge everybody in my lab, also everybody that came here. We have a really good turnout. And a special shout out to JB Friday and Susan Miyasaka, I don't know why that happened, for allowing us to have a lab space and greenhouse space at Komohana Research Extension. Thank you. This is my email address if you guys have any more questions, and that's also my Twitter handle.
Thank you, Kylie. Questions for Kylie, please type in the chat box. And our next, so we will do question and answer session after the last talk. And another talk is from Andrea Kawapata. Unfortunately, Andrea could not make it today due to her own completing other work, but she has sent me an pre-recorded presentation, today's presentation, and I will be sharing that presentation to you. So Andrea Kawapata is an extension agent based on the Kona uh, uh, on orchard crops, especially working on the coffee pest and coffee diseases. And she will be giving a presentation, pre-recorded presentation on updates on coffee leaf rust and its management. And I will be sharing that presentation shortly.
Our staff and faculty who have helped with workshops, field days, and field. I thought I'd start off with a few coffee memes.
Okay.
It goes to
Good
show
deal,
how
thank you.
important coffee is to
Okay, let's go.
people,
Let's go.
and especially
Yeah,
those
yeah.
who need a pick-me-up in the morning like I do.
So Jensen's screen. Okay. Anyway, thank you, Jensen. Yeah.
Is it working?
Working. Yeah.
Okay.
Anyhow, I'm going to start off with a couple of slides to provide a very general background of coffee production in Hawaii. Then I'll talk about where CLR is currently found in Hawaii and how to prevent its spread. Next, I'll talk about early detection and scouting efforts, and I'll give you an update on our race of rust and why knowing this is helpful. For the meat of the presentation, I'll go over CLR management recommendations, spray tips, and our sprayer calibration calculation tool. I'll close with a farm level overview of coffee leaf rust management, as well as resources available to you, including the Hawaii Department of Agriculture subsidy program. Here in Hawaii, there are nearly 1,500 coffee farms spread throughout our 6 major islands. Since last season, bearing acreage increased a little to about 7,100 acres, and statewide, coffee farmers produced 26.5 million pounds of cherry, valued at nearly $60 million. Up until about 12 years ago, Hawaii was the only coffee-producing state, though the territory of Puerto Rico also produces coffee. Did you know that coffee is now grown in California? The 50 or so small coffee farms established in the Santa Barbara to San Diego areas are likely the only commercial growing regions without any major coffee pests and diseases. Coffee is the state's #2 most valuable agricultural commodity, followed by cattle and macadamia nuts, and trumped only by seed production. Kauai coffee has the largest acreage at about 3,000 acres in Kalaheo, Kauai, and is one of the 4 mechanically harvested farms. In comparison, the average size coffee farm is just a little over 2 acres. Besides the 4 farms, all other coffee farms are hand harvested by workers, and this includes a new 12 to 1,500 acre farm in Kealakekua in Kona. Coffee leaf rust, or CLR, is known as the world's most damaging and devastating disease of coffee. It was first found on Maui in late October 2020, then in Kona, and just a year later has spread to all coffee-producing islands. This pathogen cannot be eradicated once established and is easily spread by wind and physical transfer. So to prevent the spread of CLR, we should be aware of and follow all HDOA rules and permits for coffee materials moving throughout the state as well as into the state. It's recommended that before, during, and after visiting any farm, regardless of CLR presence, that you disinfect your hands, clothing, footwear, tools, equipment, vehicles, and anything that could potentially harbor and spread CLR from one site to the next. Use at least 70% alcohol, a freshly made 10% bleach solution, and detergent, hot water, and dryer heat to kill CLR spores. Early detection continues to be extremely important for farms that do not yet have CLR, as all commercially grown varieties in Hawaii are susceptible to rust. While there is a working group to address the importation, rearing, and distribution of resistant varieties, they are not yet available for replanting, and so we need to monitor for CLR incidents. Systemic fungicides have not yet been approved for use on coffee in Hawaii, so research emphasizes the need to detect, monitor, and control CLR at a very low level of incidence of less than 5%. At a level higher than 5%, spraying with current contact fungicides is ineffective. CLR can easily be missed when scouting, so knowing where to look can be helpful. To detect an initial infection and in areas of low infection, try to check as many trees as possible for rust. Early in infection, coffee leaf rust is typically found on leaves on the bottom third of the tree canopy. Observations in Hawaii also indicate that CLR is found early on in areas where overstory trees shade coffee and where vehicles, pigs, workers, and other people frequent. If you're unsure if what you're looking at is coffee leaf rust, take a few photos and send the photos to me or to the Department of Agriculture. We can help you distinguish between coffee leaf rust, Cercospora leaf spot, and other problems. There are currently over 50 races of rust in the world, and researchers from Portugal recently identified our rust as race 24. From the 10 samples that were analyzed, data appears to show that this was a single introduction to the state. Race 24 affects many of our commonly grown varieties, and literature on this race shows it to be prolific, quick to germinate, infectious, and aggressive. So knowing this, we will need to be aggressive in our management efforts and find varieties that are resistant to this particular race. Depending on severity, spray applications, and environmental conditions, it typically takes about 30 days from initial infection to when CLR lesions produce spores on the underside of the leaf. Unfortunately, our rust takes a shorter amount of time from infection to sporulation, so spraying will need to be more frequent. Along with early detection, coffee leaf rust management requires a whole systems approach that includes good field sanitation. Maintaining weed and ground cover control around the coffee trees reduces humidity and the ability for CLR spores to splash back up to coffee leaves from grass, weeds, and seedlings during rain. Improving and maintaining tree and soil health is also very important. It's recommended that coffee leaf and soil samples are conducted at least annually to better understand tree nutrition and soil pH status. These results can help with fertilizer adjustments and to mitigate deficiencies and toxicities in the soil and plants. While coffee root knot nematode is an ever-present root pest for Big Island coffee farms, including foliar fertilizer applications and increasing soil microbe and organism diversity with composting, mulching, and the use of ground covers can help growers keep coffee trees as healthy as possible. There are 4 main pruning methods conducted in Hawaii, and coffee pruning, or at least desuckering, is recommended on a yearly basis. Overall, pruning and desuckering is done to remove excess, older and less productive verticals and laterals, to remove infected leaves and branches, and to encourage new growth. By opening up the canopy and allowing better airflow and sunlight penetration to the branches and base of the tree, you reduce humidity and freestanding water, which helps CLR thrive. Other benefits to pruning and desuckering include increased yields, reduced labor and costs with stumping and block stumping, reduced pest and disease pressure, and the ability to spray and harvest easier and more effectively. For coffee leaf rust management, consider removing the lowest laterals to increase airflow and sunlight to the ground and to help with spray coverage, weed control, and fertilizing around the base of the tree. Depending on whether your spray frequency and how long it takes you to prune, try to either spray right before or right after pruning to reduce the spread of CLR spores. Keep infected materials on site and mulch, chip, pile, tarp, and keep covered for at least 6 weeks. Because of CLR and the extent of infection on most farms, I'm recommending that growers stump a third of their farm in large sections and then selectively prune in the other 2 sections. Block stumping will help by clearing large areas of CLR-infected leaves, as well as berries with coffee berry borer. Coffee leaf rust will be minimal on the new growth on the stump blocks, and growers will be able to regain control and restart with healthy verticals. In the section that will be pruned at the end of the current 22-23 season, keep up to 3 second-year verticals per stump or a combination of 3 first and second-year verticals and prune all other growth. This will alleviate the field of verticals that are bent over, too tall to harvest, and less productive. In the section that will be pruned at the end of the 2023-2024 season, leave up to 3 first-year verticals and prune all other growth. These verticals will give you 2 years of production before the trees are stumped. Fungicides are an important tool for growers, and rotating approved fungicides in different frac groups is highly recommended to prevent pesticide resistance. Currently approved contact fungicides will kill spores on the outside of the leaf, but they do not kill CLR lesions in or on the leaves. Under an EPA Section 18 emergency exemption, coffee growers can use Preaxorzemium, a translaminar fungicide that with good coverage and proper rates can provide up to 21 days of leaf protection from germinating spores and new infection. A rotation of these contact and/or translaminar fungicides must be applied when CLR incidence and severity is under 5% for effective management. Past this 5% level, spraying along with pruning and removal of infested plant materials may be necessary to regain control of the disease. While research is still needed to determine the best timing, rotation, and rates of currently approved fungicides for CLR control, below are a couple of the various possibilities of CLR fungicide rotations based on the different frac groups. There are organic products within each frac group, so rotating between copper products, Serenader double nickel, and oxidate will help an organic farmer maintain their certification. If including Preaxor into the rotation, a farmer could spray oxidate followed by the first application of Preaxor, then copper, a second application of Preaxor at least 30 days after the first application, and then Serenade and copper again. Preaxor cannot be sprayed within 45 days of harvest, and only 2 applications are allowed per 365 days. When spraying any pesticide, please read and follow all label directions. Cover up with the proper personal protective equipment to protect yourself from any health hazards while handling, mixing, and applying pesticides. As you are aware, pesticides and labor are expensive. Under and over applying pesticides can also have legal, environmental, and financial repercussions. Calibrate your sprayer to ensure that proper pesticide rates are used and that the application is effective in managing the pest or disease. If you need a refresher, please join us for sprayer calibration field days this coming March. Most fungicides can be tank mixed with Bovaria, but tank mixing with oxidate or Preaxor is not recommended or allowed. Lastly, including a surfactant with pesticides can help obtain better coverage by allowing water droplets to spread and cover the leaf surface. The following spray tips may help you get better coverage and control of coffee leaf rust, as well as with other pests and diseases that require spray application. Open up the canopy by removing low-hanging and excess laterals and verticals so that the spray solution can get to all berries and leaves. CLR lesions produce spores on the underside of the leaves, so be sure that the fungicide spray is directed to the undersides of the leaves as well as the top sides. Use Surround WP or water-sensitive paper to help determine if you are getting good spray coverage throughout the tree. If you are not getting good spray coverage, consider focusing on the quality of the spray application and not speed, and consider using other sprayers, wands, nozzles, and pressure or aperture settings that may help you obtain better coverage. And whenever making a spray application and any modifications to spray equipment, calibrate and recalibrate your sprayer. Calculating rates of pesticides can be quite daunting, and so we've created an Excel sheet that helps to eliminate hand calculations and reduce error. After calibrating your sprayer, all you need to know and enter into the spreadsheet is the product, the rate per acre, and the spray tank volume. All other numbers will either be carried over from the calibration worksheet or calculated automatically. You can download the Excel spreadsheet and supporting documents at the hawaiicoffeeed.com/sprayer calc website. Coffee leaf rust is new, and so it's a learning process on how to best manage this disease. I'll go through 6 points to try and bring it all together in a summary. Managing new pests and diseases is expensive and can be devastating to businesses and industries. We should all help to prevent the spread of coffee leaf rust by following sanitation practices and quarantine regulations. Growers should be monitoring for initial infections and spraying early on in infection for more effective management of the disease. Fungicides in the different frac groups should be rotated to prevent pesticide resistance, and spraying should take place at least every 20 days to curb spore germination. Prune in large blocks to reduce the amount of infected plant materials. To encourage new productive growth, increase airflow through the trees and farm, decrease excess humidity and shade, and improve spray coverage and make other farm activities easier. Throughout the year and as needed, maintain weed control, remove unwanted coffee seedlings that could harbor CLR, fertilize the trees, and mulch and compost to improve and maintain tree and soil health. And lastly, don't forget to incorporate coffee berry borer control in with coffee leaf rust management. I encourage you to visit my hawaiicoffeeed.com website for up-to-date information on various coffee pests and problems, CBB and CLR information, ag announcements, and other coffee-related info. If you hover over the Events and Announcements tab, it'll pull down a listing of our upcoming events. Also, if you're in the coffee industry or know folks in the industry, our Hawaii Coffee Industry Needs Assessment and Coffee Leaf Rust Survey is still open. We welcome your feedback and have the survey translated into Spanish as well. Please don't forget that the Hawaii Department of Agriculture's Coffee Leaf Rust Fungicide Subsidy Program is up and running. Those who apply and submit receipts can receive 50% back on the cost of fungicides with reimbursements up to $600 per acre. There is a cap of $6,000 in reimbursements per farm, and this is in addition to the CBB subsidy. Please contact Melanie if you have any questions about the program and if you'd like to sign up for it. And that's the end of my presentation. Thank you so much for the opportunity to share this information with you. Again, I'd like to thank all who have provided support to my extension program and my outreach efforts. And if you have any questions, requests, concerns, please feel free to contact me. My contact information is there on this slide. Thank you and have a great day. Bye.
Thank you, Andrea. So if you have questions for Andrea, you can email Andrea or also put the questions in the chat box, which I will forward her in a few days. Thank you, Andrea. Thank you, Jensen, for taking care of this presentation also. So, so you can put the questions in the chat box for Andrea as well as Kyle. Now for Jensen, the next presenter. So next presentation is from Jensen Uyada. Jensen working together with Kunhui Wang on current invasive pest concern of avocado lace bug. It came to Hawaii some year ago. It has been spreading in the different parts of the island, in Hilo, in Big Island, and also on Kauai. And this has been one of the most asked question in this invasive pest mini conference, in the two previous mini conference. So it's my pleasure to bring Jensen Ueda and the team. Jensen is our edible crop extension agent based on Oahu, and he likes to do a small scale trials on the current invasive pest concern. And today he will be presenting avocado lace bug management in Hawaii. Jensen, please, whenever you are ready.
Thank you, Roshan. Let me pull up the presentation. You see the full screen?
Yes.
Okay. So I guess avocado lace bug is a pretty new pest, and I think everybody has seen this probably on their avocado trees. I know heavily on Oahu as well as the Big Island, you're starting to see a lot more of the symptoms. And the reason why this project came about with Dr. Kunhui Wang, her lab, and myself, we were getting a lot of calls from homeowners, commercial growers on ways to control it. And I don't think there was a lot of information at the time. This was about 2 years ago, on what growers could do to prevent the damage to their trees so that they could get avocado fruiting. So I kind of did a search to what products were available for growers in Hawaii that was registered on avocado. And from that list, we kind of chose products that we thought would be most effective against the avocado lace bug. And that's how the project kind of got started. So for those of you who don't know what avocado lace bug is, it was first reported on Oahu in 2019, and it's now pretty widespread throughout Hawaii. If you look at those images on the right, you'll see a colony of avocado lace bugs with adults, nymphs, and eggs on the undersides of the leaf. And typically, it'll affect trees of various ages. It does affect pretty much all of the varieties of avocado that we have in Hawaii, but different varieties have different levels of infestation. So some trees will completely defoliate, and other trees will have all their leaves, but will be have minor damage to them with this chlorosis. In severe cases, it'll become brown necrotic and then typically fall off. Once those leaves fall off the tree, you'll typically see a lot of the fruit fall off as well because there's not enough leaf to support the fruit load. These insects typically have a 3-week life cycle in Hawaii. And because of this, we have multiple generations a year. Eggs are laid in pretty loose rows on the undersides of the leaf. And you can see some of the tar-like material that's surrounding the eggs, which kind of protect them from beneficial insects. So it can be a challenge to keep these under control naturally. And because of this, we've decided to kind of figure out chemical ways that we can reduce the populations on trees across the state. So what we did was we kind of created a scale on level of infestation, and we use that as our metric sort of for observing some of the chemical treatments that we were doing. So in this image is an example of a healthy tree. Typically that included no infestation, no damage, would be a healthy tree. If there was some initial damage where you start to see a few leaves here and there that had infestation or initial egg production, we consider that on the 2 on the scale. If we start to see that expand out, we consider that a 3 on the scale. Pretty severe spread throughout the whole tree would be the 4, and then complete infestation and leaf drop, we were considering a 5 on that observation scale. So the main objective of our project was to evaluate a series of different insecticides, both organic and conventional products, and their efficacy on the control of avocado lace bugs in Hawaii. We also want to evaluate the efficacy of the organic versus chemical insecticides in a rotation, because pesticides have a quantity of product you can apply per year, as well as a resistance that can build up by the insects to certain chemicals. We wanted to see if we put the products in a rotation, how would that affect the efficacy of the control? So this research was done at the Pomoho Research Station on Oahu. In our avocado collection, there's about 36 established plants within that field. So the first trial, we included 11 insecticides and an untreated control. Each insecticide had 3 replicates, a replicate being an individual tree. And then we spray them at 2-week intervals. And then we tried to get coverage at a rate of about 220 gallons per acre. We included a surfactant. I think Andrea mentioned in her presentation that the use of surfactants could help increase the spread of insecticides, fungicides throughout the leaf surface because there's a waxy cuticle that has high surface tension. So we chose kinetic as our surfactant that we used in our trial. So each product got kinetic in the application. And then to apply the products to each tree, we use a backpack mist blower. In this case, it was a steel SR450 backpack mist blower that holds about 4 gallons on your back. And then we get into the products that we selected. Some of this was based on the work that was done by UC the UC colleges on avocado lace bug and the publications that they put out. And then some of the other ones were products that we thought would be effective on a true bug like the lace bug. So Admire Pro was one of them, Ecotech, Entrust, Malathion, Moltex, Movento, Impede, Microtrol, Pyganic, Sivanto, and anything with the asterisk is a organically approved product. And in this case, we had one RUP or restricted use pesticide product, which is the Mustang. So these are the products we decided to spray each individual tree with just the product to see how effective they were as a standalone product. So the chart here, you see some of the results, and on the left, you can see that rating scale from 1 to 5. So the pressure was pretty high initially of the experiment. And then we used that as our starting point. And then from there, we did the applications. The dates you see at the bottom are collection dates. And pretty much most insecticide sprays at the 2-week intervals pretty much reduce the damage of ALB throughout the orchard across all products. Some were more effective than others. Like you can see the Admire Pro was one of the higher end products when we were doing some of the data collection. And then if you look at this chart, it's an average rating scale over all the sample periods that were taken. Entrust, Malathion, Mustang, and Pyganic kind of reduced the damage scale of ALB, which is kind of hard to see because once the leaves are damaged, you can't really undamage them. And then we also collected some data where we're counting basically the percent dead lace bugs on each leaf. We selected 5 leaves per plant and replicated them 3 times per treatment. And pretty much all insecticide treatments resulted in more ALB death than the untreated control. You just had some that were a little more effective. So the top being the 100%, you had Admire Pro being pretty good at all collection dates, being close to that 100% level.
You had Sivanto Prime was also pretty good at the beginning, kind of tapered off towards the end. Initially, when we did this, the Ecotech seemed to be pretty good and was one of the organically approved products that you could use. And Malathion was also a pretty good product, as well as Pyganic was pretty good. You see some drops here and there for some of the other organic products. Like this is Moltex here, Impede, and Trust. They all seem to be more at the 75 percentile range. Sorry. And then average of across the 4 sampling times, it seemed like Admire Pro, Ecotech, Mustang, and Sivanto Prime were most promising in causing higher percent of ALB deaths on the leaves. But you did have some of the other products still getting at least 70% control or 70% dead after application. So getting to our objectives, we wanted to see that rotational product. Now that we found out that pretty much all the products had some level of control, we wanted to see if we put those into a rotation based on the amount of times you could apply that product per year, what that would look like. So in this case, we used the same trees. And this is the proposed insecticide rotation that we kind of came up with. And this was based on number of applications, expected time of flowering for some of the products that you couldn't have on the plant during flowering, or you wouldn't want it at time of flowering just because you don't want to interact with the beneficials as well as the bees that come in for pollination. So that's why we came up with this schedule. So the schedule would typically have to change depending on your trees and when they tend to flower. But this is kind of what we came up with based on what we thought would work. So our second trial executed on April 9th and went till September 17th at the same collection at Pomoho Experiment Station. We applied foliar sprays within 2 to 4 week rotation. We had chemical, and the conventional rotation had both organic and non-organic products within that rotation, just to fill in some of the gaps. And then our organic treatment, solely ARMI-approved products that we could use in that rotation. And then we had an untreated control. We utilized 30 plants, 10 plants per treatment as our replications. And then data was taken every 2 weeks. So the chart on the right, you can see we kind of deviated from the schedule a little bit, but for the most part, we followed that schedule for the initial rotation period. So over that 5-month spray rotation study, the chemical rotation resulted in the lowest ALB damage, though the organic treatments also had some control. It was slightly lower than that untreated control. So if we look specifically at the conventional treatments, in terms of percent ALB dead on the leaves, the chemical treatment resulted in the highest ALB death, whereas the organic sprays had an intermediate mortality rate compared to those conventional products. If you can see here, that Admire Pro application, pretty much immediately after application, you can see that increase in amount of ALB dead on the leaf. You also see those after the Sivanto application here on that September date. And then malathion also, you can see that it kind of maintained a level of control. In this case, we learned about danitol, I think, at this time. So we included danitol as a last minute product into the chemical rotation. And danitol is another synthetic pyrethroid similar to the Mustang, which was reported as having really good efficacy from the UC school information. And then Ecotech was applied as that organic product into the chemical rotation to kind of fill in the gaps where we couldn't have enough products to cover some of the other months, which had some mixed results within that particular treatment. And then we also got a sample of Pure Crop 1 at this time, and we're trying to see how well that product would work in our rotation as well. So looking at the organic side, the program started with that Molt-X, and you can see that it's kind of trending down after that application. The Ecotech kind of had a little bit of maintaining some dead. And then after the Pyganic application, you see the spike in a number of dead, which was expected by the Pyganic product. And in this case, the Pure Crop kind of helped maintain the level of death or percent dead on top of the leaves during that period. So overall, you're not going to get 100% control, I think, with the organic products, but it'll still be better than not applying anything at those points. I think frequency of application is important with the organic products, and then getting good coverage. So making sure that you do really good pruning practices to keep the canopy short and open so that you can get the product underneath the leaves where they need to be. And then as frequent as possible. I think 2 weeks, every 2 weeks is a reasonable time, but I think you can go up to a month and still get relatively good control. And you have to stay on the population to make sure it's not continuously spreading to the orchards. So I brought up that Pure Crop One product, and we heard good things about it. It's basically a soybean oil and oil nanoparticle product. Helps penetrate into the cellular wall of the insect and disrupt their enzymes. Right now, I think we have a mixed idea of whether or not that product works really well, but it seems to be doing something. So that's why we were including it into our particular trials. And then that Danatol product, which I mentioned, we included based on that UC information, is a really good product, but it's an RUP product that unless you have a license, you won't be able to apply. And then I think looking forward, because there's a level of resistance or tolerance to infestation by different varieties, I think looking forward, it may be more practical, at least from an organic standpoint, to identify varieties that are more tolerant to the damage, meaning they'll still get damaged, but they still can produce relatively good yields with that damage. And there are a few trees that we found get lower amounts of infestation, and some that are really heavy. So I think documenting those varieties and seeing if it's similar across the state as far as level of infestation for specific varieties is an important thing that we need to look for in the future. And then to kind of summarize the information, you want to make sure you follow all pesticide label rates, restricted entry intervals, post-harvest intervals, and making sure that it's registered on the crop and in Hawaii. For most of the products, you don't want to spray during flower bloom. There are going to be a lot of honeybees foraging, and then you don't want to be contacting them while they're foraging. So being cognizant about that when you're spraying, time of day and time of year when your crop is in certain stages. Softer contact types of insecticides such as MP, Microtrol, Moltex, Pyganic, Ecotec can provide temporary control when infestations are low, probably only targeted on limited stages. More frequent sprays might be needed. And then you want to take into consideration that there's a 3-week life cycle of ALB, so timing of application is pretty important. A lot of these products have to contact the insect, so you don't want to be spraying when they're in the egg stages. Imidacloprid or Admire Pro, for example, and malathion have been really effective as far as controlling ALB and are not restrictive products, so should be easier to access for most people, but do have issues with flowering and bee control or bee damage. And there are ways you can apply the Admire Pro through soil drench. We didn't look specifically into soil drench application. Everything we did was through foliar application. That made it easier for scheduling a rotation. If you went with an Admire Pro application of in a soil drench application method, you would have 1 application per year versus a foliar application, I believe, was 4 applications a year. So it allows for extended control if you do the foliar application, but sometimes it's hard to apply pesticides foliar. So if you can do it as a soil drench, it might be easier. I think some of the UC University of California information suggested it can go up to 17 weeks to 24 weeks of control. I've been trying to treat some of the trees now with a soil drench application to see if it actually goes out to that far for some of our heavily infested trees. And then avoid use of broad spectrum persistent insecticides like carbamates, organophosphates, and pyrethroids, because they can also harm some of the natural enemies. And these natural enemies include lacewings, predatory thrips, and egg parasitoids, which might not be known in Hawaii. And then, to acknowledge some of the team from Kunhui's lab, Sabina, Lauren, Sarah, and Donna for the help, and then Department of Ag and Dr. Amjad Ahmad for some of the funding to help with this research, as well as CTAR Hatch and Plant Work monies. And then here's some information on the green and brown lacewings that can be found in Hawaii, and some growers are starting to see them more frequently now in their avocado fields, but it doesn't seem like it's a very efficient predator, but I think more research needs to be done to kind of hone in on that. And I quickly wanted to put in, so the backpack mist blower is a pretty accessible tool for applying products to these fruit trees, but it can be a challenge for some applicators because you're carrying about 4 gallons of water on your back. One, it's not enough water to cover most orchards, and two, it's kind of dangerous having 4 gallons of water on your back, especially if you're a smaller person and you're not on flat ground. There's tendency to tip over and fall down. So I kind of came up with a DIY backpack mist blower setup that you could make for about $600 to $700, which is the cost of an individual backpack mist blower. And this particular just detaches the tank from the blower itself, which is basically a leaf blower, and then connect it to an external tank, which you can pull around on a quad or ATV. And just a quick video on what it might look like. I haven't seen a mist blower before. This is kind of what the application would look like. Making sure you're wearing the proper PPE because a lot of that liquid does come back down on you. So making sure you're covered up properly. And if you want more information on how to build one of these, you're more than welcome to reach out to me. I can definitely either do another workshop later on how to build one or send you some of the information I do have. And I guess that's it. So if you have any questions, I'd be happy to answer them more.
Thank you, Jensen. Jensen, I have a question.
Yeah.
So it is this survey plan recommends spraying of some insecticide twice a month all year round?
Yeah, so I think for the conventional products, most of them were once a month. And then for the organic products, I think we're recommending twice a month. So every 2 weeks, you would go within that month, and then you would transition to the next product for the next month. And that was just to minimize the development of resistance for the organic products. And then for the conventional products, it was more because each product had anywhere from 2 to 4 applications per year. Where to spray them all up front. After that, you wouldn't have any good products to control for the rest of the year.
Okay, so if it is organic, it's a for year round, right?
What is that?
If it is an organic insecticide, it's year round. If it is a chemical, then just whatever is in the label.
Yeah. Well, I believe all the organic products don't have a maximum volume per year or per crop season, and the crop season would be annually. But the conventional products have a quantity of product that can be applied to a tree per year, if that makes more sense.
Okay.
So theoretically, you could use, say, Impede every 14 days or every 7 days on your avocado tree for the whole year. If you have the ability to spray that frequently. But for the conventional products, you couldn't spray those products as frequent because you can only spray so many times per year.
So if we spray, suppose it's a organic insecticide, do we have any indicator when to stop? So you spray, you control the insect, yeah? So maybe there's no more insect, so you stop, yeah? Is there any indicator when to spray?
Yes.
Okay.
So I would suggest that you do field scout your fields prior to application to make sure that there is insects there to spray. So you're not having to just spray every 2 weeks. But yeah, so after you do the application, you can monitor your leaves. So like what we're doing, look on the underside of the leaves and you can see the actual insects there kind of wandering around. So if you see stuff still moving around, then you're probably going to have to continue your application. When you have the new flush of growth every season, you want to make sure that you're monitoring once the population starts building up on your trees that you start your application period. So you might not have to spray in those early periods when the new flush of growth comes out until the infestation comes into your field.
Okay, thank you. So it's 12 o'clock, but we will still go take a few more minutes, maybe 10 more minutes to answer questions. If anybody wants to leave, it is free to leave, but we will spend some more time to discuss some questions. Jensen, I have another question. So
Okay.
avocado tree might be very big, yeah? So
Yeah.
do you recommend to spray a whole tree or only some branches which is infested?
I was spraying the whole tree because you can't see how far up. If it's really far up, you can't see if there's an infestation on that particular area of the tree. But I would suggest you improve your pruning practices to bring that tree canopy down so that you can access it with your spray equipment. I think for homeowners, the recommendation, if you can't spray, there is a imidacloprid product you can get from a garden shop. I believe it's a Bayer Advanced Fruit Tree and Citrus product is on the label, and it has avocado use on that label. And that product allows you to drench the soil, so you don't have to spray the tree. And then that product, I'm not exactly sure what that label specifies as far as how long it persists within the tree, and how long the control is and how long how much applications you can make, but it's available to homeowners for use. And it's equivalent to that Admire Pro product that I was talking about. Some
Okay,
homeowners can spray their trees, and their trees are 50 feet tall, so spray application might be out of the question. So that might be another option if you're willing to use a non-organic product on your tree.
So Jensen, there is another question. Have you tried to measure impact of ALB, avocado lace bug, on yield of avocados other than sunburn on fruits and stain? Is there any indication that yield mass is influenced from avocado lace bug?
We haven't done any yield data collection, but typically what I've seen from my trees is that once the leaves start dropping, the fruit drops with the leaves. So if you have a serious infestation where you're starting to get leaf drop during the fruiting period, you'll probably see a lot of those fruits drop off as well. If you have minimal damage to the leaves during the fruiting period, you'll typically see the fruit stay on, and it may be more correlated to the pollination and the fruit set based on that time. But that's just anecdotal. We don't have any actual yield data collection from infestations. And that's probably future work that needs to be done. Like which varieties typically can handle infestation and don't have severe yield loss.
Okay, thank you. So any question to Andrea will be forwarded to Andrea via email. And
I see some more in the chat.
okay.
Thing said, what impact does rainfall have on the effectiveness of the sprays? Typically, the insects on the underside of the leaves. So if you can get the product under the leaf prior to the rainfall, you shouldn't have too much issues with like product washing off because it's on the underside. Some of the products like Admire Pro, Sivanto Prime are systemic insecticides. So what that means is they can move through the plant. So once it's contacted the leaf and has translocated through the leaf, the rainfall won't impact those products. So I would say the rainfall doesn't have too much impact, I think, on the products. I think some of the organic products don't have a very long persistence on the leaf. So as long as you're not spraying in the rain, I think it should be fine.
There is more questions, so I will read this one for you.
Okay.
Does the lace bug attack certain varieties more than others, like varietal preference? Do they have variety preference?
Yes, there are some varieties, and I don't have that data yet, or that information yet on which varieties are less susceptible than other varieties. But I know some of the trees that we're seeing, you see San Miguel is a variety that's been kind of tolerant to the infestations at our station. I think the variety is called Yamagata is severely impacted by ALB, where you'll see no leaves. It'll be a skeleton. So we'll get no fruit from that tree anymore unless we spray it. So there's heavy impact of variety, I think, on infestation frequency. It's just, I don't know if it changes throughout the state for certain varieties, or even within the orchard, if some varieties are more resistant than others. So I think hopefully that data comes soon. But we don't have that right now.
Okay, so cost of spray per tree or per acre. Do you have some estimation?
No, I don't right now. But one way you could do it is measure the size of your field, and if you take that spray rotational program that we put together, you can quantify how many dollars per acre, and that's something I can generate moving forward, is utilizing that rotational program and the recommended frequency of application. We can come up with per acre numbers, definitely based on specific number of trees per acre.
Are there any organic soil drenches? Maybe no.
Not that will affect avocado lace bug. There's not that I'm aware of. I don't think there's any organically approved products that are systemic, which is required for the soil application to be effective. So with Admire, the plant will take up that Admire or that imidacloprid and translocate it throughout the plant, so that when the insect feeds on the leaf or sucks on the leaf, it'll ingest the chemical, and then that's what impacts the insect itself. I don't think any of the organic products will do that systemically. So the only way to use them is to physically contact the insects itself. So
announcement. February is the month of Hawaiian Invasive Species Awareness Month, and the presentation and events are listed in this website. I have just put it in the chat box. Our another Kylie wants to share here. So this one already answered. Kylie is around still?
I think
no, she left.
Okay. So did you answer this? Does spraying plain water with high pressure hose help? No.
It may help dislodge the adults, but I think because the eggs are encapsulated in that tar material, it won't be knocked off with high pressure water.
Well, what about pulling the leaves off? Is that beneficial or does that hurt?
I mean, if you pull off the leaves, you're going to be pulling off every single leaf of that tree by the end.
Yeah, because we've got some in like the 2 to 3 stage.
I think if you have neighboring avocados that have it, it's a matter of time before they gets fully infested, because they can move, I think, pretty far. So I don't think removal of leaves is a practical, especially if you have more than one tree.
What if you're a homeowner with one small tree?
I think you can probably prevent the spread initially by pulling off those initial leaves, but towards the later stages of that flush, you're going to be pulling off everything off that tree typically if you're not spraying anything.
All right. Thanks for your great work.
More questions for Jensen. Any preliminary recommendation on resistant variety?
I think only one I know of right now is that San Miguel, which seems to be pretty tolerant, just because I see that tree every day and I've been eating that fruit for the last 4 months, 3 months. But hopefully, I think my plan is to put something together for Hawaii on what trees are most resistant, and then from there, I think quality of the fruit that comes from those trees has to be looked at too, especially for commercial growers that are looking for shelf life, and I think shippability. So being able to ship it is an important thing for the larger scale grower. So I think that's a bigger project than I can take on, I think, right now. And if you want to send me information, I'd be more than happy to consolidate it. If you have trees that you're noticing are really heavily infested and not spraying it, and the leaves tend to defoliate the whole tree, I think that would be pretty good information. And I think that's my plan in the near future, is to put together a survey to kind of get that information. I'm not sure if anybody else is doing that right now, but that's kind of the plan.
Okay, another question. So systemic insecticide will move through the plant and it will be translocated into fruits also, yeah? There is some.
That part I'm not completely sure. My assumption is that if you're following the post-harvest interval and the application instructions, it shouldn't be an issue by the time of harvest. I think there's a 25-day PHI from application to harvest for soil drenches. And I believe the harvest interval is 1 day for the foliar application, I believe. I don't have that information off the top of my head right now. But yeah, I don't have specific information to tell you whether or not it is or is not being translocated into the fruit. So I think that's part of the scheduling that I did, was to make sure there was no systemic product being applied during flower and fruiting. So that was part of why you would fill in other products during those times of the year. So during the fruiting period for a conventional rotation, you can use some of the other products that won't persist, I guess, in the plant and are pretty more knockdown stuff. You're just trying to contact the insect and keep the population low so that there's enough leaf surface to generate energy to get good yields.
So is the organic pesticides are also systemic? Is the question.
No, I don't think any organic products are systemic that I'm aware of.
Okay, I think that's the question here in the chat box.